With that in mind, my name is Bruce Beckloff. I'm the CEO of YellowDog, and I think I would be remiss, since you made such an effort to come out and see us this morning and drink the coffee and have some croissants, not to tell you about the great things about YellowDog and what we do. Now, as you can tell, I'm an American, so it will be a bit of a sales pitch. Just grin and bear it. We'll get through it, and then we'll get to actually hearing about how people are actually using AI and ML in life sciences. I'm joined by a team, and it's an expert team. We're based in Bristol. There's about 15 of us in the room. You have the founder who's going to leave a fireside chat after this. He's the brain and the creator of the technology, Simon Ponsford. We also have our CTO. Actually, they're all grouped together weirdly. I don't know why that is. We've got our CTO, Alan Perry here, our COO, Niall Kennedy, Laura Kiley, our sales and alliances, and Ryan Proctor, our head of marketing, who's really been able to make this event happen. And we really appreciate all the effort that he's put into it. So what are we doing? So we're describing ourselves as enabling coherent compute, and because we're really trying to bring to bear what we actually do. So what do we call coherent compute? It's a simultaneous processing of a workload across on-prem, across hybrid, multi-cloud, any region, anywhere. So any resource, anywhere. And what YellowDog does is enable you to process your workloads across that coherent compute. We're an AI-enabled scheduler using this coherent compute to optimize specifically for speed, cost, and simplicity. It's a very simple product. We're a middleware layer. We sit in between apps, whether it be an AI app, whether it be a life sciences app, engineering app. Really where we've seen a lot of uptake is in financial services, where you have big workloads that need to be processed. And then on the other side of YellowDog are the resources. And on the resources, we're just that thin layer to schedule and orchestrate between those two different entities. You can API integrate to us very easily through our SDK. We have a standalone user interface if you want to stay out. But with that API integration, you can seamlessly connect to YellowDog, and you really don't even need to know we're there. We're just there to orchestrate. We're agent-based. So what we do, we put little YellowDog agents on the resources that call back to the platform to let the platform know what resources are available. Well, what that means is, one, you get to understand everything that's available. But also, if you have new resources you want to make available, if you're a large biotech company, you have a new on-prem or a new data center you want to bring online, you simply plug that into the YellowDog platform, and the platform immediately knows it's available. So essentially, we give you a single pane of glass to manage all of your resources, regardless of where they sit, whether they're on-prem, cloud, or even what region. We're battle tested. We've been out for a while. A few examples here. One example was with OMass Therapeutics, who some of you know. We enabled them to do a year's worth of drug discovery in a day. In fact, we actually enabled them to process 337 million compounds in 7 hours. And although that's an amazing ability, what that really translated to in the economic benefit, they were able to save $5.5 million annually by bringing all that compute and being able to narrow that window of that drug discovery. The other big one we did was with AWS. We built the 6th largest supercomputer out of AWS publicly available compute. So it was publicly available on the 4th of July. We did 3.3 million virtual CPUs. We deployed at about 8,000 virtual CPUs a second. So we built it in roughly 50 minutes, and then ended up breaking it down in two. Why is that breaking it down important as well? Not only so we shut down, so you had no more lingering costs after the process was complete. We also work with startups. We work with Elvijs, who's doing, they're actually making a digital twin of a heart. We enabled them to do 1,000 virtual patients out of the cardiac data from 75 patients. That took about 45,000 hours of processing. It was done over 7 days, but still quite a lot of processing for a startup to be able to comprehend. And also in the AI space, in a slightly different one, one of our partners, Mindtrace out of Manchester, doing asset inspection, sort of enabled two things for them. One, they've got the fire forget model where their customers are able to send their data to Mindtrace, and we help Mindtrace just send back an answer. But sort of also most notably for startups, we enabled them to go multi-cloud. So not only enabled them to get the best pricing across multiple clouds, but also secretly use the cloud credits for multiple clouds, as you all the startups in the room might know is there. So again, there's multiple ways that we're used. We go big, we go cost efficient, but it enables customers to really optimize their workloads for their business rules. It would be naive to think that we're the only ones out there. There's a lot of competition. Here's probably the most popular ones. We have another list of probably 30 on a separate table. And if you looked at the ticks in the green boxes, we all look the same, but it really isn't. We're not all the same. If we could put an overlay of ease of use across this, and people go, oh yeah, ease of use, but you don't really understand that until you get into the inner workings. But if you just look at the raw hard data, you know, we give the coherent compute, so access all your resources immediately. We provide 200 times more processing power than our nearest cloud competitor. So for big workloads, you can get through them quickly. And we're 30 times faster on the provisioning. So you're only paying for the compute resource that you are using at the time. What we're introducing today is Nextflow, YellowDog for Nextflow. And this will be our first introduction and an integration into the life sciences space. We're looking into other spaces, clearly AI. We really want to get feedback from customers about what integrations are important. Clearly, the name on the frame these days is PyTorch, and it's something we're looking into. But on Nextflow specifically, what are we enabling? We're enabling multi-cloud and hybrid, and we're the only Nextflow executor which enables multi-cloud deployment. We enable Nextflow users to optimize for their preferences. So that means you set your business rules. Your startup, clearly cost is going to come into play. Your GSK, you need it fast, you need it now so you can get your drug out 2 months earlier and generate another few billion of revenue. So we enable you to make those decisions. We are a carbon conscious company, and we enable you to actually look at the carbon efficiency of the compute you're using. We give an eco score to all compute resources in the cloud. And so you can start making your decisions within the platform to go for the carbon efficiency over maybe some other options that are available. And with us, again, we're automatic. So you press a button, we scale up, we scale down. Once you set your limits, YellowDog enables you to do it. And also, again, we enable the single pane of glass. So no longer do you need to worry about how you're doing your computing or where you're doing your computing. You can concentrate your team on the innovation part and really leave the computing part and the workload management to us. And we're also announcing today we've had a long-standing partnership with AMD. Thank you very much again with AMD for co-hosting the event with us. And they've enabled us to provide you, the users, the app providers, access to the YellowDog technology for a year for free for those qualifying. So if you're interested, this will enable you to use our coherent compute. You'll also get access to our team, our tech team, to help you get up and running quickly. And we're able to get most people up and running anywhere from an hour to a day. And again, if you're interested, there's a lot of us around, so please find us. There'll be another networking session after, or you can go to yellowdog.co/get started. It will give you access within an hour. So anyway, get in touch. With that, I told you it'd be painless, it'd be quick. That's me done. I'm going to hand over to Simon and Daniele to talk from AMD to talk about processing in life sciences and AI. Then I'll hand over to Andre and Elvijs to talk about some actual use cases of where ML is being used in the life sciences space. And after that, we'll have some time for more coffee. And look, if you need a Peroni or a Prosecco, I'm sure I can go around the corner and find some. So anyway, thank you very much for coming. And I'll hand over to Simon to take us through the next section.
very much, Bruce. Right, we just grab a couple of microphones. Daniele, please. Hello. Good morning, everyone. It's a strange trying to have a fireside chat without a fire, but we can try anyway. So thank you, Daniele, for coming along today. We've got some, I think we've got some questions for each other, but hopefully we put together some questions that you'll all find of interest. And really, I think we want to understand really about AI, ML, life sciences in the context of the people that are here today. And really how new innovations, in some respects what AMD are doing and also what YellowDog are doing to really be able to help people accelerate their scientific discovery. So, just go with you, Dan?
Yeah, that's good. First of all, thanks for inviting me today, and thanks all of you for coming and listen to this great event. So, my name is Daniele, I work at AMD. I'm in charge for market development for our accelerated computing business unit for Europe, and particularly focusing on commercial HPC and AI. So, my role is really to understand what's the need of this community and try to bring your request to our product management, make sure we have the right product for your needs. So I'm very happy today to be here. I will try to share the few things that I know on the topic and try to learn as much as possible from your question and your interest here. Thank you.
I should also say as well that Orion's over there on the side. He's got a microphone to be able to go around as well and be able to ask, if people really ask any questions, etc. We're very focused, very happy to so if we kick off, is it okay? So obviously there's been a lot of conversations in the last 12 months or so about AI, ChatGPT, etc. And AI seems to be coming into everyone's lives today. But with regard to life sciences, how can AI help?
But this is very good question. You're right. I mean, ChatGPT changed everything. So we used to work mainly with scientists on traditional HPC, and since end of last year, basically the entire market changed. We saw an incredible increased demand in GPU, incredible increased interest in this kind of technology. Even my mother started talking to me about ChatGPT. So you see that things really went mainstream. So probably there are two main things that we noticed from a demand perspective. First of all, we start seeing a lot of demand from a lot of new industries that usually didn't use HPC technology. So everyone wants to benefit from the large language model, from generative AI, and this is opening a lot of new discussion with new customers in new vertical that we were not used to others. So the market is becoming really big and bigger, opening many new opportunities. And on the other side, we see that traditional scientists using to benefit from HPC system, performing scientific simulation, are start using AI and ML side by side with traditional simulation to expand their capability, to have quicker answer, to have another way to achieve the results. And this is common to basically all the research fields, and in particular for life science as well. We've been working, in example, on the AlphaFold model, where we really got very interesting results on our platform. Depending on the model, between 75 to 150% compared to the competition, and this is really opening a lot of new discussion and new interest from many areas.
I guess for most of us, using computers for years, until a couple of years ago, we hadn't really heard of accelerators. We had GPUs for gaming, etc., but now they're coming into normal computing workloads. So what's really the advantage of using an accelerator?
Well, if you look at the CPU and the GPU and you look at the specs, typically a GPU consumes twice the power of a CPU, more or less, and can perform up to 20x compared to a CPU. So if you are really targeting to achieve very large computing power. In example, if you're targeting to deploy an exaflop system, GPUs are the only way to go. I mean, you don't have any other option, because any system that's not based on any sort of accelerator would consume too much energy to be practically usable. And that's why if you go to the top 500 list, which lists the fastest computers on the earth, basically most of them and probably all of the top 20 are based on GPU accelerated system. So this is really key for scientific advancement, because it enables something that you cannot simply do with just CPU. Of course, it also brings some challenge, because you need to port your code to GPUs. You need to enable your code to move from CPU traditional platform to accelerated one, and you may have different provider for the acceleration platform. So you may be sure that your code not only run on AMD accelerators, but also on NVIDIA accelerator, Intel accelerator, because you want your code to be portable and to be usable on multiple platforms.
Okay. And obviously, there are all these accelerators around, and most people are probably familiar with traditional AMD-based GPU cards and PCs and laptops, etc. But obviously, you've got some new GPUs. There's some are already available. Others, I guess, are coming into play at some point. What's the advantage? What are the key things that a scientist here would be enabled through an AMD GPU over potentially another vendor GPU?
So the main thing that we want to bring here to the market is diversity. So until today, you just had one option, which is great. But now we see that there is the market size and the demand to bring alternatives to the market. So our platform is very performant in terms of pure hardware potential. If you look at Frontier, which is the #1 system in top 500, reaching 1.2 exaflop of performance, it's based on the MI250 series from AMD. So you have very high-end performance on the hardware side. But we also believe that one key element is the openness of the software. So we want to make a researcher free to move between platform, to move to AMD platform and from AMD platform to other platform if there is a need. So all our software stack is open source, there is no charge for that, and it's designed for portability. Just to give you an example, our programming language is called HIP. If you program with HIP, basically the code that you run can be compiled for both AMD and NVIDIA. Okay, so it's really designed with portability in mind, and that's a key element that we think we are bringing to the market. And talking about this, I have actually a question for you on this topic, because you see there are many different platforms, right? And one of the key advantage of YellowDog is the ability to have a single entry point to an entire world of computing. So how do you handle these different platforms, this hardware diversity, and what's the key advantage of using YellowDog over direct access to cloud provider, for example?
Sure, I think it's a simplistic way of looking at YellowDog is a single API to the entire cloud. So you can talk to YellowDog and it will translate that and be able to work with your cloud accounts for the entire cloud, and also to be able to address on-premise as well. So we find a lot of people, particularly research institutes, have their own in-house compute, and they want to be able to absolutely maximize the use of that. And with the YellowDog agent, they're able to do so. And so it's not a case of whether I only run in the cloud or if I only run on-premise. You're able to be able to maximize what you've already invested in. And then what you've got is you've got all the cloud accounts. And most organizations will have a cloud account, and they want to be able to make use of that. Like you say, we'll talk about new accelerators. Most people are not going to have the latest AMD accelerator, but it will probably be available in the cloud. And so you're able to use YellowDog to be able to say, I want to run these jobs on my CPUs on premise, and maybe these on my on my NVIDIA GPUs, but I've got some other things that I've been designing. Maybe I've been using PyTorch, etc., and I know that's going to work. That particular job's going to work very, very well against an AMD system. YellowDog can go out and can actually identify and find those systems as a complete view of the entire cloud. Continuously we're pulling down the information of every single instance type, the price of every instance type, the availability of every instance type, which data centers are in terms of how it's going to work. And so YellowDog can actually go quickly pick that and deploy. And come across lots and lots of startups. And the usual story is we're a new company, so we decided to go with cloud first, and we started building something. Maybe we built the first version of our software, say in AWS, and we've targeted a particular AWS data center, and now we're going to build one, and now we're ready to run. And then the first customer comes along and says, I don't want to be able to use that yet. I don't want to use a data center. I want to use a data center in another region. So a lot of people make a mistake of probably just targeting a general cloud provider. They target particular regions and particular data centers. They realize they need to change their code in order to do that. And then they make code changes, and they are able to then work against another region. And then the next customer comes along and says, but I prefer to use a different cloud provider. And so every single time they come along, they find they have to re-engineer to be able to work with different cloud providers. And so YellowDog gives you that common interface that allows you to be able to work with all cloud providers without having to recode everything. If the cloud provider introduces new hardware, it's instantly available with YellowDog. So there's no change of any code. If a new generation of a chip comes out, or if there's something that's more performant, again, YellowDog will automatically adopt that, so you can automatically get that better price performance, or better speed, or better value. So it just allows you to do that one integration and be able to keep up with everything that's going on. So I think that's a real advantage of going with YellowDog over than just integrating with a single cloud provider. Of course, all the APIs update, and you need to keep working to update to keep going with those, whereas YellowDog is being do up in the background and keep up.
That's great. And what about security? Because I guess that most of these data are quite sensitive, especially in the life science field. So how do you handle this on these deployments?
Right. Yeah. So in terms of security, YellowDog actually works with the customer's own accounts. And that's probably that's important. It's not the data, etc. is not going through YellowDog's accounts. What we're doing is acting on behalf of the customer with their own cloud accounts. So data only goes where the customer wants the data to go. So they would have already signed up, maybe they've got an account with Google or with AWS and with Azure, etc. And they need to actually have images there, and they need to be able to have a network in those accounts. So what YellowDog is doing is acting really as an orchestration platform, preventing things from essentially only running there within your account. In terms of data transfer, you can transfer configuration information, those sorts of things, through YellowDog. But the important thing is, for most customers, is that their data is only going from their premises direct to their cloud account. So you can think of YellowDog as an orchestrator rather than something that's actually getting in the way and actually data being funneled through them. So really, YellowDog is as secure as working directly with the cloud provider. So we mentioned in that PyTorch, etc. Do you have any examples of how PyTorch in particular, because I know AMD's put a lot of focus on it, and PyTorch is the buzzword at the moment in terms of working with AI technologies.
Yeah, it is indeed. So we are a member of PyTorch Foundation, first of all. So we fully support this framework, and it's particularly important for us because 90% of the conversation with AI customers, basically, we understand this is used by the far majority of any AI customers. So we put a lot of effort, particularly in the last year, to support standard machine learning framework, and this with the specific goal of making easy for ML users to port and to move between different platforms. So PyTorch is fully supported. If you are using PyTorch, if you have PyTorch code running today on GPU, you can very easily move and test the AMD platform using basically the same code that you are using today. And there is a nice blog, actually, you can Google on the internet on Mosaic ML. And these guys in the US, they wrote about their experience in porting their PyTorch code between the GPU they're currently using to AMD. And the message is very clear. It was very straightforward, very smooth transition to the new platform. So yes, that's definitely something that's on top of our priorities that we are actively supporting and contributing to the new.
I just looking at YellowDog this morning, I did a search for AMD, and of course I found lots of AMD GPUs across all the cloud providers, but I didn't actually find a single AMD GPU with any cloud providers today. When is AMD accelerators going to be available in the cloud? I know you can buy them from premise, but and like you say, you've got Frontier, breaking the records with with exascale, but when a normal human's going to be able to get hold of these accelerators?
This is the big question. This is a lot of work we did this year to enable these on the cloud as well. So we will announce the new generation of our GPUs later in December. Probably if you Google MI300, you already find all the specs on the internet, but these will be formally announced in December, and we will announce cloud availability in the same time frame, and you can expect to have a number of cloud providers during the next year to make this technology available on public cloud. Cannot share names right now, but make sure. But if we do this again next year, we will have at least a few options out there. So yeah, be a bit patient.
Okay, thank you. I think you had some other questions as well to ask about the other platforms.
Yeah, so we spoke about ML,
yes,
and how this is changing a bit the hardware side of the story. And I'm curious to understand, what do you see from your side, how this new demand for machine learning workload is changing the way people use YellowDog, or what do you have to do?
So I think for us, certainly the last 12 months or so, the availability of GPUs has been a big problem. This is generally the NVIDIA's. And people come on to us and they come almost desperate and say, well, I can't configure the cloud provider, I'm trying to get hold of GPUs. They're trying to get hold of GPUs on spots, and they're finding it almost impossible to get hold of GPUs on spot because they are so highly utilized. And also people are coming along and saying, I want 20 GPU cards and I'm only managed to get 5 or 6, and I can't actually get my way out my workloads in time. I saw an article last week where some people in Korea have actually given up in Korea on the NVIDIA cards just because of availability, and are trying to actually port code to go back to running on much slower CPUs just to be able to get work done. And I think the thing about YellowDog is because we've got this view of all of those GPUs across all of the cloud providers, you can put a requirement into YellowDog and it will find them for you. And it can essentially be able to bring those together in a way you couldn't before. So if your workload, for example, is distributed and you want 20 cards and you can only find 14 in one data center, YellowDog will go and find either a data center that has 20, or it will actually go out and try, if you distribute the job and you define that within YellowDog, it will go and find, okay, there's 2 in this particular data center, there's 4 in this region. You can handle data transfer, etc., between all of those. So that's really what we're finding is people desperate to get hold of GPUs. So people have been trying to buy them, very long sort of waiting list to get hold of it. That seems to be improving, but certainly in the cloud, availability is the problem, and I think we can help a lot of people with that.
Yeah, that's one of the side effects of the OpenAI stuff. I mean, the demand really grow incredibly, and GPU are becoming rare on the market.
Yes, it's a big problem for the people who need it. Some people have just resorted to running things on premise. with gaming cards, because some of those are more available or picking up on eBay. But I think we're over the hump on that now in terms of there's a lot more GPU available. We've got the growth of specialist GPU providers as well, so there are now a new range of cloud providers only providing GPUs and everything else. So I think certainly by Q1 next year, most people will be able to access this level of GPU that's required.
You want to open to some questions?
Yes, maybe. Right.
I think Simon's demonstrated we don't necessarily need the mic.
If the demand is globally unmatched, how are you finding those GPUs? That's an obvious question. If you're saying that the resources don't match the global demand,
So
how YellowDog is finding those GPUs to do the job quickly and efficiently?
So essentially, most people, when they build their applications or they set things up, they set themselves up in one or two regions or a few data centers, and they don't really have the scope of everything. So if you take AWS and every single data center that they have, YellowDog will be able to go and find, maybe outside working hours, there might be some that are available in Tokyo, there might be some that are available in India, but most people normally wouldn't even think of looking at those locations. And YellowDog will continuously, if you submit a job into YellowDog, will continuously look for those resources. So you might have something sat in a queue for several hours or potentially even a day or so. Yet as soon as one of those GPU cards becomes available in any one of the data centers you've got allowed YellowDog to go to, it will then add that to your cluster.
So a single job can be distributed across different data centers across the world?
Yes.
A single job?
Yes. And what we
Confusion.
when we talked when Bruce put the slide earlier and the run with the AWS, there was essentially running a life sciences molecular docking workload. And really what happened with that 4th of July run, the very large run, essentially took all of the available spot compute, so all of the unused compute in the AWS data centers in Europe, then went over and started taking everything in North America, essentially to be able to get that done. And if it hadn't have run out of work to do, it would then have spread across to the US, to the West Coast, etc. So YellowDog can be, if needed, truly global in order to be able to obtain those resources.
Thank you.
Any other questions?
Yeah, one. So what do you think of the sort of split these days of on-prem versus cloud? What's the world looking like from an AMD perspective? Is on-prem dead or?
We think both will carry on. I mean, it's not a mystery that probably the big cloud provider are the #1 market buyer for hardware in general. So that's something that we see continuing and growing. But still, there are a lot of use cases where you need on-prem hardware. I mean, the material one is security. I mean, if you're a bank or something like that, you still need to run your stuff on-prem. Even with the ML use cases, the large language model, we think that a good part of the inferencing will be still done on-prem because of security reason. You may want to have, let's say, taking some trained model, do some fine tuning, and then being able to run your query on-prem. So we think that both these will keep going. We don't see the on-prem going away.
Question. So we're looking here at AI and life sciences, and if we look at those two groups, the AI world has been traditionally for the last 20 years very much cloud native. Because of the nature of ML, it's highly parallel. So bog standard ML can just run on GPUs, so it's really great for the cloud. The life sciences world, you mentioned, they've been very focused on simulation as engineering, those kind of worlds. And they traditionally, as I understand it, had a bigger focus on HPC with its tight integration of CPU, maybe GPU, but also the interconnect and the storage. Where are those two? I've seen, particularly with the life sciences world, they're picking up a lot of ML. Now, they could have two separate worlds where they do simulation and ML. But are you seeing any kind of conversion, particularly with the AI world, looking at the HPC world?
So from an hardware perspective, I come from an HPC background and I see AI as a use case of HPC, basically. So
well, that's what I'm interested in. What is it that would move somebody that, let's say you've got an AI startup, that they would look at HPC versus just looking at a regular cloud scale with GPUs?
So let's say that HPC brings some additional needs from hardware perspective. On HPC, you care more about networking, in example, about fast message passing between nodes, while on AI, you may care more about memory per GPU, because especially with the large language model, you need to host a very large amount of data for every run. With HPC, you are maybe more interested in higher precision computing like FP3264, while with machine learning, you may be more interested in reducing precision. So there are slightly different in terms of the hardware platform. We think that each of these two fields, we can optimize for each of these two fields, and that's why you see we are announcing two different line of GPUs in December. One slightly more targeting HPC, the other slightly more targeting AI, but basically you can run AI and ML on both of them, so it will depend from the specific case of the customer which of the two you want to use. But we definitely see an overlap between these two, and we see, I mean, with machine learning, you can make a query and have answer very quickly, but you need to understand if those are correct, especially when you do scientific research. So I think this is a bit of the challenge that we see from the researcher's side, and go to great.
Yeah, thank you. And I'll probably add to that. If you go back, say, about 3 years, there are almost no HPC instances in the cloud. And that's certainly changed. And usually, previously, just more commodity compute, and you didn't have the specialist interconnects, etc. And then all the cloud providers have been building HPC capacity, and a lot of that is around AMD, etc., and getting those interconnects right. And it's not, very often in the cloud, we find it's not the users are not the traditional HPC users. A lot of people, traditional HPC users, are still on premise. A lot of them haven't moved away. The people that are making a lot of the use of this hardware in the cloud are the ML companies. So they are the ones that are often taking those HPC instance types, finding that they are just, they're very good and they're available for running those types of workloads. So they're close to optimization. Obviously, perfect. If you bought your own in-house hardware, you might have a few customizations that were mentioned here, but we're finding there's an awful lot of people who are using these instances. So they're actually getting, just within the YellowDog and being able to see their availability. We could see in the last 18 months or so, the availability of those reducing because they're busier because of those ML workloads. I think it's really interesting. A lot of the people don't actually think that they're looking for HPC because they don't come from an HPC background. And that's, I guess, where people would say come into YellowDog and they would describe the type of workload they want to run. And YellowDog would find them HPC instances, but they wouldn't necessarily go into and realize they're HPC. They're just delivering those workloads within a certain time span.
Why is YellowDog approach for cloud storage? Because we see with cloud providers, they often put egress is always a cost. So once your data is there, you are not incentivized of running somewhere else. So you're trying to be constrained about that. So with YellowDog, you have access to multi-cloud. What do you do with massive storage?
So YellowDog has essentially we have an object store service allows you to be able to work with all of the cloud providers using a single API. So we support all the storage types in all the different cloud providers. But as you mentioned, you can end up with a situation where you have all your data in one cloud, and then of course you're getting egress costs if you want to push it to another cloud. So it might be that you've got or even the same cloud in a different data center, you might have different costs. So at the moment, YellowDog mainly optimizes around the best source of compute. So that's very, very CPU focused. What we're working on at the moment is the best source of data. It's very often better to actually bring the compute to the data rather than being able to essentially try to shift data to the compute. So we've essentially got this model that we're working on essentially to be able to work out what's actually the cost of being able of taking that data somewhere else, whether it's an egress cost, maybe there's an inter-region transfer, and also the time it's going to take to move it there. And by using machine learning to understand about the availability of instances, you can make smart decisions on it. No one can overcome, I guess, the egress costs. Those exist, but you can start to make intelligent decisions about where to run, not just purely be focused on that CPU or GPU and how much that costs per hour. So I guess that's our approach to it. We're also creating something called our data mesh service as well, to be able to make sure that your data can be made available wherever that compute might need to run. And also we've got something as well to be able to predict when you're going to start to need to run a workload. So you might be pushing data somewhere, so it's only when that data's there you're going to start to run the compute and start to consume. It's a big problem, and YellowDog isn't going to solve it, but I think we're certainly ahead of the field in trying to work out ways to reduce some egress costs, and I think there's a holistic view of the overall cost and the overall time to deliver that job based on all the different elements and not just one. Okay. Well, thank you everyone for the questions. Thank you, Stella, as well.
The mic works up there as well, but behind the screen.
What?
The screen? Or this one works.
Ah, okay.
Just have to have a booming American voice and you'll be fine.
Okay. Hi, everyone. Hi, my name is Andre. Yeah, I think we can start here. So I'm from Panakeia, and I'm going to be giving a tech talk today about some of the solutions we use for using AI for cancer diagnostics, and the title specifically is Optimizing for Scale in this field, like molecular-driven analysis. So a little bit about me first. So I come from a research background. Before joining Panakeia, I did 8 years of experience in research. My PhD is in surgical robotics. So I was basically trying to develop robotic tools and robotic devices to improve the outcome of surgery, try to make it either more precise, safer, and better for the patient in general. And then in 2021, I decided to shift to the industry, and I was looking for companies that had a research DNA, and I found Panakeia. And so I've been there for this about 2.5 years now. And then right now I am the technical lead there. So it means I am the lead of the engineering team. And my main responsibilities are the development of our internal platform and the productization of the ML models, so we can deliver those to the patient, to the customers. So this is our team. As you can see, we're still relatively small. We have only 7 developers at the moment, spread between engineering and research team. Engineering team focus much more on the infrastructure development, software engineering tools, and productization, while the research team focuses a lot on actual model architectures, training methods, and data strategies for the ML development. And then, of course, we have the business, product, and regulatory team taking care of regulatory requirements for medical applications, product development, and customer outreach. And we also have 2 in-house pathologists that help us developing the ML models. So they help a lot with incorporating biological knowledge onto the development of the models, understanding applications that are more interesting, and helping us decide which scientific approach makes more sense from the biological point of view. So we have better chance of making correct predictions with the models. So this is the outline of the presentation. I'm gonna start by presenting a little bit more what Panakeia does, telling you about what's our approach for biomarker profiling and how do we tackle this keyword that is multi-omics. Then I'm going to talk a little bit about the technical challenges that we have with this problem, what kind of engineering issues we face because of the problem we're trying to solve. Then I'm going to go into some of the key things that we do to tackle these problems. This is going to be a very high level presentation, but I hope there is time for us to discuss whatever is most interest of the audience. And then at the end, just a little bit of future vision, some of the next problems we hope to face very soon when things start growing. Okay, so I would like to start with this diagram, which is quite dense, especially for someone that doesn't come from biological or medical background. But basically, what this shows is the current workflow that there is the pathologist in cancer diagnostic. So you have a suspect of cancer, you take a biopsy, your tissue becomes, you make a slide of the tissue, and that goes to the pathologist to analyze. They make the primary diagnostic and they confirm it's cancer. So they request wet lab tests to characterize the biomarkers that are present in that cancer. And that information is going to be very important because it's going to let you decide what kind of treatment you have to do. So depending on the hospital and the infrastructure and logistics involved, this can take 1 to 4 days. And if the result is positive or negative, great, you can make your decision. But if not, you may request subsequent testing, and that may be more complex tests. They're not only more expensive, but take more time. So for example, for breast cancer, a common test for HER2 can take up to 2 weeks. And if you need genetic sequencing for more rare mutations, you can take 2 to 3 weeks to get that. So while you're waiting for all these tests to happen, the patient is there waiting for her treatment, waiting for the decision, and time is passing. So this can be something that is very problematic. So the approach Panakeia takes is we go back from the very first H&E slide where the primary diagnostic is made, and we train ML models that are allowed to detect the biomarker status if it's present or absent directly from the morphology of the cells, and we can give that answer in a couple of minutes. So that means you replace the entire need for wet lab testing, you replace this 2 to 3 weeks that a patient is waiting, and you're able to make a decision right at the moment of the primary diagnostic. So not only reducing costs, but accelerating accessibility for treatment for the patients. So we really are a digital, medical digital assay that analyzes the H&E stained image of the tissue and gives you the presence of the biomarkers. And we actually work with different kinds of biomarkers. So you can have like genomic alterations, so what kind of mutations are present in the DNA of this patient, proteomic expressions, so proteins that may be over or underexpressed, alterations in the transcriptomic profile of the patient, all of that directly from the H&E, from the morphology of the H&E cells. And how can we do that? Well, there is a biology dogma that tells genetic alterations in the DNA translates to alterations in the RNA expression, which change the protein expressions, and then in the end affect the metabolite pathway. So basically, all of these levels of alterations have an impact on the phenotype, which is the morphology of the cells. So that what that means for an ML point of view is there's a signal there to be seen, right? So if you know how to detect, and if you have enough data to train your models, you can actually detect that signal and even go back to the very first level with your genetic alterations, identifying DNA mutations and so on, directly from the morphology of the cells. So that's, you know, it's known that it's possible, but it's still very challenging, and it's something that's very hard to validate because the signal sometimes is very faint, and you need to train the models to be able to pick on that.
Just a very quick question.
Sure.
So is it you predicting these as like outputs, or are these the inputs into the model?
So the model analyzes the images and predicts the status of the biomarkers. Of course, for training the models, we need the ground truth for a lot of those, right? So we have a bunch of images with the corresponding genomic status of alterations. We train the models to learn that signal, and then once the model is trained, we can apply that to unseen images, and that gives you the status of the biomarker.
And in terms of the transcriptomics, is that really spatial transcriptomics, or are you doing?
Right now we don't do spatial. I'm gonna go a little bit more in that later. But yeah, you can tell like these genes are overexpressed or underexpressed in terms of RNA transcription without spatially localizing in general on the slide. Yeah. So yeah, so the ability to predict all that, replacing those tests and making them more efficient, and a lot of very interesting consequence of that is one of the problems with multi-omics, you're trying to predict lots of different genetic alterations, RNA transcriptomics, and each of these tests require a little bit of tissue, but in the biopsy, you only have so much, right? You only collect a tiny amount of tissue from the patient. So sometimes you may want to run 20 tests, but you may only have enough tissue for running 10. By going fully digital, our assays don't consume any tissue, right? Once you have the image, you can test all possible available biomarkers. So you don't have to worry about destructing tissue, and you can always, repeat that and run the test later with different models and so on. So the approach that we are providing here is truly a multi-omics platform, where from a single image like this, that a tissue, you can say, what are all the different models that I have available, all the different biomarkers I can predict with this, and you can run all those models at the same time in parallel to have a full comprehensive multi-omics of the status of the patient. So just to show a little bit of numbers and the power of this, we validated this approach with a systematic review study, where we identified more than 4,500 different biomarkers over more than 30 different cancer types. And this was done on only 10 weeks of compute time. So in our servers running all these different models in parallel. And we have a preprint that we are publishing now in a major journal showing that that even for this wide variety of biomarkers, there really is a signal to detect. Of course, some of them are easier to predict and others are more challenging, but it's really a sound approach that makes sense from the biological point of view. So that wrapped up the biology part and what we're trying to do. So what are the technical challenges of actually making that work? So first of all, based on our experience, we learned that there is no one-size-fits-all solution. So you can have models that try to predict multiple things together, but truly, if you want to get good performance, it really comes down to fine-tuning the specific models for each biomarker, because the signal in each of them is going to be tied to different parts of the morphology. So you really have to define models that are specific for one biomarker and one disease indication. The second challenge is that the performance requirements are extremely high. If you want to deliver something that's going to be used to diagnose a patient and decide the treatment, you really have to have very high sensitivity, very high specificity, right? And with ML, it's very straightforward to do something decent. It's really hard to do something excellent. So when the performance hires are really high, that makes things way more harder. And yeah, as I said, there's a wide variety of biomarkers and disease indications that you may want to predict. So how do you go about that, right? So how do you get something that is very customized with very high performance to this wide variety of biomarkers? That's the real challenge. Now, in terms of technical limitations from the more engineering side, one of the problems then is you need high amount of compute and also man time involved on this. So for each application, you may want to do hyperparameter optimization, try different combinations at the same time, try different models, architectures, and especially if you start incorporating biological knowledge, you may want to do that on a case-by-case basis. So a lot of man time involved as well, and a lot of compute. The two things go hand in hand. Second problem is there is the high data heterogeneity. So since we are working with multi-omics, these biomarkers we predict, they come in very different formats. So you're trying to predict a genomic biomarker, you can have a list of genetic alterations or even raw DNA sequencing. But when you're talking for protein expression, then it's like different levels of quantification of expression. And each one of these biomarkers in the wet lab, they come from different tests that come from different machines and different vendors. So there is no standard format to represent all of that. You have to handle these different types of data. The images themselves are also comes in different formats. So right now in pathology, there is no standard format like in radiology. They have DICON. For pathology, still relatively new. So each scanner outputs images in their own format and they don't talk. So you have to figure out how to access all that if you want to support scanners that are used in many different hospitals. And also you have very different quality standards in terms of the staining and the scanning quality. So depend on different labs, they have different reagents, they have different machines, calibration. So images can look very different, even if they are from same disease, depending on which lab they came from. So things that we have to overcome. And then finally, something that's also very interesting. Fortunately, cancer diagnostics is a very dynamic research field. Lots of new treatments coming every day, new biomarkers being discovered every day that can impact the treatment. And if you really want to be involved in this research aspect and being able to collaborate with this, you have to be able to deliver results very quickly. So that's something that we're very engaged. All of these new drugs being developed, lots of them require a companion diagnostic test to be able to profile that certain biomarker. And we at Panakeia, we want to provide solutions for that as well. So that means when we come to a pharma customer that's interested in detecting a specific biomarker, we need to be able to develop a new model for that really quickly. So the structure of our platform, of our development platform, has been focused on all of that, overcoming some of the challenges in limited compute time, dealing with this data heterogeneity, and also being able to deliver proof of concept results really quickly, so we can engage in conversations on how to streamline the development of one specific model for one specific application. So now I'm going to go to some of the things we do. First, let's get rid of the basics. All of that is very standard, so I don't think there's any surprise here in terms of ML development. So we use Python and PyTorch for most of the backend and the development of the models. Our application is mostly a backend because we integrate it directly into the PAC systems that the pathologists work in the hospital, but we do have some front ends for internal use and also for marketing purposes. We use React and Redux for those. For CI/CD, we have Jenkins running our builds on edge of our GitLab pushes. Then the packages we deploy, we have our own PyP server. And then finally, for actually deploying the solution, we use Kubernetes as orchestration. So our solution is multi-container. We have several Docker containers for different parts of the pipeline, which allows us to load balance a little bit. And we have Kubernetes as the orchestration for that. Now, a few of the more interesting things we do. This may also not be entirely surprised, but there are things that we've done that kind of like define the way our platform is structured, and they work really well for us. So I'm going to go a little bit into the details of these. So the first one is the design of our database. So our database, we have a relational database at the core of our platform, and the key decision we made is that it is connected to everything, right? So for ML, strictly, you don't necessarily need, you have your data, you have your status, you can train a model for that. But we use the database for our data lake. So all of the images, all of the biomarkers, all of the multi-omics data that we have for every step of the model development, validation, and deployment. So every job that you run leaves a trace in the database. So everything reads data from there and saves data there. And also lots of our internal automation tools also communicate with the database and track the progress there as well. So what you have in the end is a database that is fully traceable and reproducible. So if you lose the model, you can easily reproduce that because you know exactly which data set was used to train that, what training parameters and everything. So that makes it really powerful because it's very flexible for us to work. Another interesting aspect is that we have many degrees of abstraction between the biomarker and the prediction task. So for example, the biomarkers are integrated as they come. So you have the genetic alterations, the HER2 tests that I mentioned for breast biomarkers. They come as they go, but the task that you may be interested in predict may change. So for example, for certain cases, you may want a binary classification at positive and negative. For certain diseases, you may want more granular information. You may want to find an intermediate value. So what we have by separating these things in the database, if you want to predict a different task, you don't need to change your data. You don't need to change your training pipeline. You only need to change the task definition. So you have this abstraction layer that allows you to use the same biomarkers and the same training pipeline, but just defining different tasks to approach different use cases. So that actually gives you this effect that from the training point of view, anything is a biomarker. As long as there is a task defining that this combination of data is positive and this is negative, it's treated as a biomarker. It does the training classification from the ML point of view. Another interesting thing we have is a very modular machine learning pipeline. Again, this is also a no-brainer. The more modularized your code is, the better for development and deployment. But I want to point to one specific division that we have is between our pre-processing and the training pipeline. So when I talk to pre-processing, it's basically just reading the whole slide image file, which these files can be huge. Each image file can be several gigabytes in size. So reading that, patching it in tiles, and then applying color normalization or some transformations in a way that we have data that is treated and ready to be used for training. And this typically can come at the beginning of your machine learning training job, but by separating the two things, what we allows us to do, reuse the same pre-processed data sets for multiple training jobs. So you want to try several different model architectures, you want to try several different parameter optimization. You can do everything without having to repeat the pre-processing every time. So that saves a lot of computing. And then in terms of platform, we do a mixture of on-premise and cloud. So we started mostly with cloud-based solutions, but I think everyone that works with cloud, especially in a small company, knows it's going to be quite cost expensive, very, very expensive. So we have our on-premise server that ends up being most cost effective for most of our development. And we use cloud as a complement whenever we need more compute, whenever we need to deliver results quicker, whenever it needs to scale certain experiments, we can get extra computing in the cloud. So that means the entire pipeline needs to be set up to run in any platform, right? So it doesn't matter if it's a server or if it's a cloud instance, it's in AWS, GCP, the entire code runs everywhere. And then finally, also a no-brainer, we have mostly recently introduced job scheduling and automation in terms of the entire pipeline. So it's basically a scheduling system that handles all these different parts, pre-processing, training jobs, data analysis, configuration setup, and so on. And it runs the jobs both on-premise and the cloud. The backbone is Learn. So we use Learn for hardware resources allocation and scheduling. But we also have our custom in-house scheduling logic on top to be able to handle more complex things like batch submission. I want to submit several jobs at the same time that have similar characteristics, or job dependencies. So I want to define a chain of jobs that start from a pre-processing and then do all the sequence of steps. This can be customized in our data scheduling, and some data analysis as well. And what that is aiming us to produce is the really completely automated experimental sequence where the machine learning expert is only going to design the experiment. It's going to say, based on this problem and what we know from the biologic and from the state of the art, I think this approach makes sense. Design the experiment, hit go, and then the automation takes care of, okay, this is going to run in the server. It's going to be ready in this amount of time. So that's something that's still a little bit getting there, still in progress, a lot of work happening, but it's allowing us to move in the right direction. So these were the main things I wanted to talk. As I said, very high level. Happy to dive into more details of these, or ask about or answer about other things that we do that I may not have covered. So with this in mind, what we have today now is, although we can do pretty much everything, we're still focused on a few key biomarkers and a few key cancer types because we have very limited manpower. So our flagship device was for breast cancer diagnostics, so for ER, PR, and HER2 biomarkers. And then we also have one in colorectal and a few other cancer types that still in development. Still some manual intervention required for running all of these things. So I think that's a utopic dream of every engineering company, is to automate everything so you don't have to actually manage the repeating stuff. And still a lot of focus on incorporating this biological knowledge to help make the decisions for the ML training. Where we're getting tomorrow, with the automation we're getting for these tools, we're diversifying more the biomarkers and the ML strategies we're doing. So pretty much tackling more problems at the same time. ML expert is focusing more on experimental design and not so much on manual requiring and making sure the jobs are running the way they're supposed to. And each of these steps is generating output data that can be easily sent to our pathologists so they can actually interfere more quickly, right? So they can every day give comments and review if what model is doing makes sense from the biologic side of view. And then my final slide, just some of the challenges that we hope to face soon. One of the problems that we don't really have solutions at the moment is training on ever increasing amount of data. So, you know, in the past months, our data sets grew from a few hundred gigabytes to a few terabytes, and in very soon there's going to get tens of terabytes and so on. You know, for generative AI these days, you can have really massive amount of images. So that's a problem that we have to handle from an infrastructure point of view, especially if we remain with this hybrid approach. Increasing level of internal automation, that's something that's a dream for everyone. Everyone wants to have as much as possible of that. And then finally expanding to other data types, going beyond these H&E images, so liquid biopsies, spatial omics, some more granular information that also target different biological problems that are also still very hot topic, research topics at the moment. So that concludes my presentation. I'd like to open for questions now. Yes, please, sir.
Hi. Thanks for the presentation. Two questions. What about sort of ground truth? So obviously humans aren't one of these answers. So humans have different sort of expression. And you mentioned using your genomics. So again, correlations in DNA changes and correlation in phenotype changes. I was wondering how you incorporate that in your models. And secondly, how do you go from where you are now to deploy it onto a complex system?
Okay, so regarding the ground truth, we have models for each of the different types of omics, and we combine them. So if we're developing a model for a genomic biomarker, then the ground truth would be the NGS sequencing that was being done for that specific gene. For more clinical ones like the breast cancer, the ground truth is the wet lab test that's being done, like the IHC test. And a lot of problems we have is that these tests require human involvement, and if you take two pathologists to grade the same test, they don't have 100% concordance. So we know that we can't fully trust the ground truth. So that's some problem that we have. Even when we get ground truth, we cannot fully trust that. So we do a lot of data curation in-house with our pathologists. They also grade the slides independently to make sure which of the labels we can actually trust and which are maybe not as reliable. So we try to have a curated data set for training. And I'm sorry, could you repeat again the last part about the deployment?
Yeah, sorry, I was saying that, so now you've got all this, you go into what you do out to how do you go from there to then
Yeah, so once we have the model, then the same approach for running the preprocessing and prediction can put in the deployment stage. We have cloud approach as preferred, so we put that in a cloud instance and we expose the API server so the user can send requests, send the image, and get the biomarker status. But the preferred way to deliver that to the pathologist is actually integrating that directly in the PACS system that the pathologist is using. So in the system that they already used to work for doing the primary diagnostic, there's just an extra button, run analysis with AI. When they click that button, the system sends the image to our cloud instance that runs the ML prediction and sends back the biomarker status. So that's how we've been integrating that. Yeah, of course, you need to have a CE marking approval on all the devices. Everything has to be regulated for medical use. And you have to work directly with the PACS providers to support integration. So we've been working with some of them like Sectra, Philips. They are all building their AI stores to allow running with third-party analysis, and they have the license terms and the separation of responsibility of how that works. So the legal part is very complicated, very a lot of work on that as well. But yes, it is a lot. Yes, please.
So how big is your training set? And I presume you're using neural networks, you mentioned deep learning. How big is your neural network?
Yeah, so for the specifics of the ML approach, there is an SSL model that is trained first, unsupervised, and then the deep learning profiling models with vision transformers. I don't know the numbers of layers if that's what you're interested, but in terms,
that is actually what I'm interested in.
Yeah, I don't know the numbers. I usually leave that for the research team. But for the data sets, we can be from a few thousand images to tens of thousands right now with the images we have.
Okay, and amongst the challenges, I mean, you didn't answer the question about the layers, but what effort are you making in terms of explainability of results? So when your thing gives false positives or false negatives,
yeah. So
crucial in this.
Yeah.
What's your ability to explain what's going on?
So we have explainability interpretability tools that we use for internal development. So we can export the attention map that shows which parts of the image your model has most focused to make the decision, because you run analysis on the patches, but you don't take all of them in consideration at the same time. So the model focuses in specific parts, and we can show a heat map that shows, okay, the decision was made based on these parts. We can also show the scores, prediction scores that were predicted at each patches to make sure that the prediction is working as intended. But one of the challenges is that if you show that to a pathologist, not always is going to correlate exactly with the region that the pathologist would pay attention in the image, because they are not trained on doing prediction on the H&E slide, right? So what we've seen is that explainability tools not always help convincing and building trust, because if they don't align exactly with what the person would expect, they actually make the life harder. What we've seen that works best in terms of building trust is actually blinded validation. So you take your device and you apply that to different data sets from different sites with blinded data, and if you can really demonstrate that you can generalize in performance, that's what really builds trust. So we have partnership with several hospitals in the UK, in Wales, Scotland, and England, and they provided us some data for doing blinded validation, and that's what we did. We applied the same breast cancer device to 5 different hospital sites, and we show consistent sensitivity performance across all of them. So that's the kind of evidence that usually is more important of showing that the safety and performance of the device is actually sound. And the main reference is you have to show that it's as reliable as the wet lab testing, which they know is not perfect, which is what they have at the moment. So as soon as you have enough data to prove that your performance is as good as the wet lab testing, that's the kind of thing that is more important for decision on adopting, at least for early adopters, rather than the heat maps, because everyone expects the AI to quickly guide you through the process. But in reality, the signal is sometimes so hard to detect that even if you show the correct thing that the model is doing, that may not convince the person if it doesn't match what they expect. Yes?
So I have two questions. So in terms of training the model, you mentioned pre-processing. Do you pre-processing for training as well, or do you avoid pre-processing so that the model becomes invariant to those kind of features that can? And the second question is, in terms of the multi-omics predictions, how are they used by
Yeah, so in terms of the preprocessing, yes, a lot of the effort in preprocessing is eliminating the data heterogeneity. So with the color normalization, we use generative adversarial networks to normalize stain for different labs and so on. So the idea is that, yes, the patches that get to the training are as standard as possible. Try to eliminate a little bit of the bias and a little bit of difference that you get from different scanners, different parameters, settings when scanning the slides and so on. And then
so your hope is that the model will be able to out those, like, because obviously ideally you want a model that's invariant for those kind of features, right? Those kind of options.
Yeah, so in the SSL models, which are the backbone, you do data augmentation to try to make them invariant to anything but the signal. We also use dense to make sure that you're actually predicting the biomarker and not the lab or the scanner where it comes from. So you try to separate these two things. But when it comes to pre-processing, you actually try to get patches that are as standard as possible to make the life easier for the model. And then in terms of how do you provide results to the customer, you try to provide in the same way as the current state of the art is. So if they expect to see a genetic mutation as a list of mutations per genes, that's what we provide. If you expect to see the grading for a specific marker, this marker runs to 0, 1, 2, 3, that's what we try to provide as well. So basically, it's not something that is going to be working together with the pathologist when making the diagnostic. It's something that replaces the assay, but the information is the same.
So if you're trying to get the phenotype on an HIV slide, the results that you get from your model will be comparable to what one would get from two biopsies, or?
Yes, but we focus on specific markers. We don't do the full whole genome. We don't predict all the genes at the same time, right? We have one. So if there are 4 or 5 genes that you are interested in, we develop models for these 4 or 5 genes. So that's the difference between the NGS.
Yeah, sorry to have to stop there, just because we need to get to the other, and hopefully you'll be around for the
sure, of course.
as well, so you guys can continue that no problem. But anyway, thank you, Andre.
Thank you.
You had a few more slides.
Yeah, there was some. Yeah.
There we go. All right, Elvijs.
Okay, thanks very much. Pleasure to be here, and despite the train troubles, I'm glad I managed to make my slot. I'm sure everyone's very interested in lunch, so I'm going to try and make this relatively quick. I've got kind of 3 main objectives in this talk. I want to talk a little bit about neural engineering and why I think it's a very exciting area. I'm going to talk a little bit about the BIOS vision, so that's the company I represent, on how we think we can take advantage of this opportunity. And finally, I'll try and dive a little bit deeper into some of the nitty-gritty to give you a little bit of a tangible idea of what we do. So let's see how it goes. So let's start off with BIOS. So we're a neural engineering company. So what that means is we're trying to treat diseases via neuromodulation. We call these neural digital therapies. So we clamp onto a nerve target somewhere typically in the peripheral nervous system and try to treat diseases. So why does any of that make sense? Let's just explore that in a little bit. So the brain controls all major organs. So as I'm on this stage, as I'm inhaling, my lungs are expanding, and there's 2 types of receptors that measure how fast and how slowly and how fully my lungs are expanding. The slowly adaptive stretch receptors and the rapidly adaptive stretch receptors that then send a signal up into my brain. same time, baroreceptors, so these are tiny little pressure sensors on the arteries, tell the brain how much blood is being pumped in. So the brain then takes all of that information, it contextualizes it with the sensory information. So I'm presenting, so it's probably going to be driving my heart rate up. So this is how it kind of controls all the major processes. So there's no real question that it's a hugely important organ system. What's maybe slightly more surprising is that it's actually really, really important in almost all chronic diseases. So almost all chronic diseases have a huge neural component. One of the, obviously we don't have much time to go over it, but maybe if we just mention heart failure. So heart failure back in the day was thought to be kind of a muscular disease, and people were trying to treat it with kind of muscular drugs. And there was a huge paper released in, I think it was 1970s, and I'm really sorry, I'm forgetting the name of it, but it kind of very clearly showed the neural component to heart failure, how kind of the brain essentially drives the heart too much and essentially kills it over time. And once that was understood, from this is where the beta blockers essentially arose. Doctors understood that you need to treat the nervous system as well as the muscular failure, and this is kind of to this day, it's beta blockers are a huge part of the standard of care in heart failure. And these sort of stories extend across a number of diseases. So there's a lot of chronic cough and all sorts of neural reflexes. So that's kind of the background setting. And we're seeing a lot of interesting therapies being developed on the back of this. If you were on BBC yesterday, there was a really cool article posted about a Parkinson's treatment via an epidural implant in the spinal cord that essentially completely changed the quality of life for the recipient. So we focus on a very specific part of the of this whole area. I apologize for the poor images. We focus on the peripheral nervous system. So we don't focus on the brain stimulation stuff. We don't focus on the spinal cord. We're as a company, we're placing a bet on the peripheral nervous system. Here we're pointing you towards a very important vagus nerve. I don't have the time to talk about the physiology too much, but it is one of the major nerves in the parasympathetic, so kind of the rest and digest part of the nervous system, and it innervates a bunch of organs. It goes to the heart, to the spleen, to the lungs, etc. And just in this area, there's been two very big successes for what we call neuromodulation devices. So you have a pulse generator implanted, kind of like a sort of a clever pacemaker that slaps onto a nerve and essentially zaps it. And by zapping it, there's two things. It kind of tells the brain something that it kind of mimics the organ signal. Maybe you signal more to kind of mimic more activity to then tell the brain to relax. And equally, you can be mimicking signaling from the brain down to the organs again, changing, like speeding up heart rate and stuff like that. These are the sort of outcomes you can get. And it's really interesting. It feels like quite a crude approach, but it works. There's been FDA-approved therapies based on these pulse generators in epilepsy and drug-resistant depression, which is very, very exciting, and it led to a lot of investment in this field. However, in recent years, it's also experienced some setbacks. So I'm going to focus on heart failure because that's very close to what we do. Two failed phase 3 trials for heart failure. This is a huge deal. This represents a huge amount of investment and a lack of treatment for a very, very important disease. And so why are we wanting to go into this area, right? So if you look at the plot there, this is a very famous plot. This was behind kind of the latest of these phase 3 trials. And there's a lot to unpack. We're not going to go over all of it, but we're going to just focus on a few very small things. So the state of the art in the industry when thinking about these therapies is thinking about the zapping as kind of thinking about the dosing in neuromodulation as kind of trying to figure out roughly what intensity you're wanting to zap at and how frequently. So more frequently means that you're sort of mimicking more interactions, and more strongly means that you're recruiting more and more of the nerve target. So the nerve consists of a bunch of different subfibers that all go to different places. The stronger you zap, the more of them you recruit, and the more deeply you get into the various organs. This is obviously an oversimplification, but this illustrates how the industry to date has thought about these problems. And you can see the two axes, the X and Y axes are two parameters, so the intensity and how frequently we spin. And on the Z axis, again, you typically have some sort of a biomarker that's supposed to be a therapy engagement. So you're trying to figure out your dosing. You work with your surgeons and your clinical specialists to figure out what biomarkers you should be optimizing for. And typically this is done via animal trials. So you prove it in animal trials. You figure out, okay, so let's say 10 milliamp intensity and 10 hertz frequency is the money dose. Then you take that to your clinical trials. You adapt it as you prove safety. You also take information about how well it's working on the therapy in humans, and you adapt the dose slightly. But fundamentally, it's kind of a one-size-fits-all approach. So you have like these optimal sort of the money frequency, the money amplitude, and you're trying to essentially attain that in all humans. We believe that this is a huge part of where the trials are struggling. We think there is no one-size-fits-all solution. Everyone's nervous system is different. Everyone's background setup is different. Your general state of your nervous system, maybe you just had coffee, so you're kind of on high alert. We believe that the answer to this is essentially live dosing. Being able to live see what's happening in the body and then adapt dosing to that. That's kind of the hypothesis that we're taking, and we want to, and this is what essentially the business is all about. We're trying to validate or invalidate, hopefully not invalidate, that this works at a very high level. Hopefully that makes sense. And note how kind of in it's also it kind of represents a novel approach to this area. It's kind of really thinking about treatment as an algorithm. So that's kind of one of the big leaps that I want you to take with me. We're essentially kind of thinking about this as algorithm as a treatment. That's part of it. And the hope is that the more data we're able to extract from the human body live real time, crucially neural data, and as we'll see, that's a huge challenge later on. We're hoping that's going to be the difference. So that's what we're about. And kind of as a sort of a broader contextualization, we're broadly trying to get to one of these huge sort of index events in biotechnology. We don't think we're there yet. We're thinking we're currently trying to build out the technology, but we're aiming big. We hope that if we can make this work, then this will open up a whole new host of diseases and treatments. The human neurome is, I apologize for the marketing blurbs there. Anyway, so with that in mind now, we've kind of spoken about this high, big picture vision, and that's really great, but does it actually work? How are we going towards that vision? So let's take a look at some of the nitty-gritties. So this is kind of at a very high level, the anatomy of a trial, which is, trials are still kind of the, so the currency of the development in the field. You're doing your development via trials. So you're testing out your algorithm as a treatment. And you're also working in parallel with scientific hypotheses, because typically diseases are not very well understood. We're having to do kind of fundamental research in an indication area as we're doing this. And what you're seeing here is a cross-section of the nerve, and each one of these little holes, hopefully you can see it, that represents a different nerve subfiber, which will go to a different organ or to a different part of the brain. And these are our contacts, little blue ones, where we're, one, listening as to what's happening. Is there more activity in this region? Or maybe that's a pulmonary region. Or when we're zapping, do we want to zap more towards the pulmonary region, or do we want to go for a cardiac region? Those are the sort of determinations we're trying to make live. 1,000 micrometers, so the scale is very small. It's one of the challenges. Two, the speeds are very high. these are electrical signals, and they're not quite as fast as copper wire, but you're still looking at, in order to make any sort of sense out of it, you have to sample at something like 30 kilohertz, which, to give you an idea, that's roughly 2 million rows a minute. And high density data, because this is obviously just one of the cross sections, we have a bunch of them. So you're trying to combine all of that data live and make sensible inferences on top of it. One other interesting thing is this is a nasty, messy biological system. You're seeing all sorts of noise signals. You can see the heartbeat in your nerve signal. You can see any sort of muscle twitches. All of that is a huge problem for us, but it also, you know, if you can solve these problems well, then that's a really nice IP moat. And the way we work is we transfer that data typically into the cloud, which doesn't seem like a big deal, I'm sure, for people here, but if you're just contextualizing the industry, it typically works and you do a full day of recording and you have like some sort of a live view on what's happening. But then you literally pop it on a USB stick, you give it to your scientists in the lab, and then they take weeks to analyze it. And maybe your sensor wasn't attached correctly right at the start, which is a surprisingly easy mistake to make, because the noise is very difficult to distinguish from genuine neural signal, especially if you have 40 channels coming at 30 kilohertz live. So being able to stream it to the cloud and then connect it to our analysis teams in like various locations. So typically we run these trials in North America, and the analysis group is mostly European based. So we stream it into the cloud and then do all sorts of feature extraction. So you're looking at the neurograms here. So these are direct responses on the nerve, and each of those highlighted buttons is a different type of nerve fiber that's being recruited. So the A betas are more motor neurons, and A gammas are and A deltas are implicated in breathing, and etc. etc. What's really cool is that we're able to see that live. That is kind of, as far as we know, that's not something that's done in the industry. And then finally, we take it to the BIOS AI. I can't remember the marketing term. I'm sure there is a better one, but I couldn't remember it. And I'll talk more about that in the next slide. So that gives you a rough idea. So two things I'd like to call out here. So one is, as you see, this order of tens of gigabytes of raw arrays, that's not really big data just yet. Even after the live feature extraction, I'd say we're still in a low data regime. Why is that? Currently, the big insights on indications are being made by bespoke analysis, working closely with the scientists and with people in indication specialists to kind of tease out useful information. That's not the long-term state that we want to be in. We want to be applying deep learning and other cutting-edge techniques to gain an understanding of what's happening in the neural system, right, at any given instant. The challenges are, we currently don't believe we have enough data, and two, this is very, very noisy data. So if you think about the deep learning promise and where it's really proven its worth, it's kind of got two huge areas where it's been amazing over the last 10 or so years, and that's natural language processing and that's computer vision, but not messy, nasty neural data. So that's kind of one of the challenges that we have to make, that we have to get over. And also you typically don't have good labels, because as you're listening to the nerve, it's sort of, there's just a lot of noise, and it's very, very difficult to get good labels for it. The way we're trying to solve that problem is by collecting a lot of nerve signals ideally from a lot of different locations to build up a full picture of what's happening in the nervous system, and then in parallel, put it next to other biomarker data. So this might be your heart rate, it might be like your respiration rate, all sorts of kind of live signals. And hopefully then we can piece together a picture. We're not there yet, but we are actively thinking about kind of foundational models for neural science, and we're working with some partners in Mila, which is the Benguio's lab on these, but I actually can't talk too much about it because it's as of yet it's not patented. So hopefully that makes sense. One of the other things that I wanted to call out here is that just this simple making sure that the data is streamed out to the cloud and into the hands of scientists allows us to test a lot more different doses live in a trial. So remember, if we go back to what was this slide, I introduced this idea about how the dosing is sort of find out the two parameters, the intensity and the frequency. And the idea is that you don't necessarily know what that response surface for the biomarker that you're interested in, what that looks like in a new animal or a new human. And you're trying to figure out what's happening. So you're applying this dose for a little bit, measuring the response, and getting 1 point on the surface, etc. Typically, state of the art is you do about order of tens of those a day. So 10 points on that surface, which doesn't give you a lot of resolution, and which means you need a lot of trials to get a rough sense of what's happening. We're currently doing of the order of thousands, low thousands, but we're very excited about this capability. It allows us to bring this vision of getting the responses live and then making good decisions based on those responses. That's what the system is built for. This is what it enables. This is one of the key things that we're trying to do. Yes, foundational models are cool and they're very exciting, but in terms of the therapy difference makers, we think this fast turnaround is the really key advancement. So now, cool theory, but does it actually work? So what you're looking at here is that's a real plot for one of our trials. And what we're looking at, again, is you've got some sort of parameters that represent the dosing. And on the z-axis, you have a very specific response. That's a specific therapy response. In actuality, when we're running our algorithms, you're typically going anywhere from 2 to 9 dimensions, depending on the waveforms and the doses we're considering, and a bunch of different potential targets. And we're trying to balance them. So you have, typically what happens is you have a stronger therapy effect, but you also have a higher side effect. So it's really, really important to find these trade-offs in the dosing space where you're still getting your therapy whilst mitigating the side effects. That's what this is all about. And this is what our system is built to do. That's kind of our AI, main AI play right now. And we validated that this probably works for the indication of heart failure in the order of tens of animals. We then wrote up a very clean protocol and tested it in five preclinical trials. With the idea that we didn't want it to be just kind of a research protocol, we wanted a very strict evaluation. How do you know this is the optimal dose that you found? Are you measuring all the side effects in parallel, etc.? And so far the results are really great. We've been able to achieve the target dose. And crucially, one of the really interesting things, if you remember back in the day, I said, 10 minutes ago, I said that this idea of static optimization, we think this is not the right approach. We think that there isn't one dose that works for all. You have to do live dosing. That is something that I should have probably put a visual up of, but the 5 animals here and then the 10 research animals, the optimal doses are all different. So it's a very important validation for us scientifically that we do think that everything's indicating that personalization is the way to go. We wrote up this work and applied for quite a prestigious prize run by the National Institutes of Health, the US behemoth, and we were one of the winners based on this specific work of Neuromod Prize Phase 1. So we've kind of convinced ourselves that this is really exciting and cool. We've convinced the Neuromod Prize panel, which is really nice. Our next steps are taking this into humans and testing whether it really works when the rubber meets the tarmac. So I think that's where I'll stop, and I'll let people ask questions if you're interested, or if you want to go for lunch, that's fine as well. Thank you.
Yeah.
First trial you led the anthem update back in 2014, patients, the patient actually died, I think, from the vagal nerve stimulation itself. And some of these vagal nerve stimulations are already being used in clinical practice already. So sweat trial already. How does this kind of differentiate between both what's on the market? How do you control safety?
So the Anthem HF didn't make it to the market. So the Anthem HF is the 2013 study. So the plot that you saw there was for their next evolution. So I wasn't familiar with the patient dying. That's quite interesting. But nevertheless, the phase 3 trial was approved. That's the Anthem HF REF. And they enrolled about 500 patients into that. So I don't think, if there was a case about VNS being, vagus nerve stimulation being dangerous, I don't think it was strong enough. We certainly don't see that evidence in our animal trials, and we're not aware of it being fundamentally dangerous. Because you also have to remember that these are heart failure patients, so this is a very tough condition. So I don't expect there to be a human problem there, human survivability problem. In terms of what differentiates us, it's a live dosing. We think the name of the game is personalization. And Anthem HF REF did not really do personalization.
I'm a bit surprised you don't think safety is a problem here, because if someone's got a clot in their artery, you're stimulating the vagus nerve, do you not see a possible spread risk?
So assessment of those sort of questions is the job of early stage human trials. And if they are not deemed successful, you do not get to phase 3. So I believe the evidence points that it's safe. But yeah, I mean, it's a huge thing. So we have to be very careful. But the clinical trials area as a whole is, I think it's a very good system. If something's on the market, it's very likely to be safe. So I believe there's a question here.
You mentioned noise being a major problem. So how do you manage it? How do you manage getting rid of it?
Yeah, so mathematics is kind of the answer. So we do a lot of signal analysis, and we develop a lot of in-house IP to deal with it. It's not easy. We've spent significant amounts of time to just validate that we're looking at genuine neural signal and stuff like that. Overall, I think the area is there's a high level of integration between each stage. If you don't have a good surgeon, if you don't have a very good electrical team that works on the hardware, and if you don't have a very good analysis team, if either of these 3 pieces is missing, I think you're going to struggle to do anything in this field. You really need all of these 3. We have invested heavily in, typically in, if we look at our competitors, they invest heavily in the first 2, the surgery and the hardware stuff. They tend to be very, very good at that. We believe that it's a slight competitive advantage for us to also realize just how important data analysis is, and we've been investing heavily in capabilities there.
And sometimes if you allow some noise in the system, it prevents overtraining model. So do you sort of question if that you allow some noise, or do you?
Well, it's not up to us. The noise is there, so we have to live with it. Do I think it's a problem for models? Yes, I think it's a huge problem. We're still kind of, we're as you can see, it's not even easy to define a learning problem here, right? Like, what's your ground truth? Are you trying to kind of ping up when you see an action potential signal traveling along the nerve? Are you talking more about, like, have we integrated our stimulations into the target organ? Like, what's your learning problem? Do you want to predict heart rate based on what you see on the nerve, right? Like, you can slice and dice this in many different ways. And in all of those cases, yeah, we believe that noise is a huge problem, and we're just in early days of kind of, we're now at a stage where neural networks tend to outperform our sort of simple baselines, simple white box baselines, but this is just early days. Yeah.
But I mean, if I put together what you said,
Yeah.
medical science is uncertain, the data is noisy, and the model is inscrutable. So you're just hoping that the outcome is, in some probabilistic sense, comfortable, and give up understanding.
No, that's just the neural nets. So this is not a neural net. These are probabilistic models. We want explainable stuff. We're taking this, the end user of this will be, we don't have a license to practice medicine, right? We have to produce something that the surgeon will be happy to work with. So it has to be very, very explainable, which is why we're using typically Bayesian optimization, Gaussian process sort of techniques for this. The neural nets, that's more of those are currently moonshots. I do not know how we take that to clinic just yet, but we think it is a piece in the puzzle if we are to realize the bias vision, bringing neuromodulation to all diseases.